jump onto our second presentation. Cary?
Is that showing up?
Yes.
Cool. Yeah, thank you everyone for the very interesting and in-depth presentation on sort of the bigger picture and a little bit of a dive onto the peptide manufacturing portion. I am going to be focusing exclusively on that tiny circle, which is the peptide manufacturer, specifically personalized manufacturing and how that might differ from a regular large-scale commercial peptide manufacturer. As Trishul mentioned, I'm Cary Potochnik. I used to lead our neoantigen manufacturing process, joined Polypeptide to work on that program, and sort of have been with it since early development stages through the opening of our GMP lab and until very recently managing the production of neoantigen polypeptides. So brief disclaimer, we are a publicly traded company. I do work for Polypeptide. There may be forward-looking statements in this presentation. It is not an invitation to purchase securities. If you're going to buy them, please do that. If you were not, please do that. Thank you. So I'm going to run through who we are, who's Polypeptide, the history of the neoantigen manufacturing at Polypeptide, some challenges that are associated with that personalized approach, a brief shill on why you might want to partner with a CMO if you're moving into this space, and then we'll have some time for questions. So overview of who Polypeptide is. We are a leading independent peptide CDMO. Recently, this has expanded to include oligos, and we're also willing to look into other things if it sort of fits into our manufacturing expertise. We're a full service provider for complex peptides using different technologies. So whether in the neoantigen small scale side, that may mean very small automated manufacturing systems, or on the other side, potentially very large automated manufacturing systems. And if the program doesn't quite fit into those, we've got a fairly solid custom manufacturing for both peptides and oligos that uses some automated systems and also relies on manual operations before a process can be fleshed out enough to be automated. We have a global presence. We've got manufacturing sites all over the world. Innovation leaders for sustainable and peptide-based drugs. We ideally for us want to be with you from the development of your product, help you with the process development, move that into a larger scale manufacturing system, and then continue through until it's a commercial product where we do have the scale to continue producing that for you. And a high quality workforce, an international management team. We've been around for a while. If the team at Torrance hasn't seen it, likely someone at one of our other sites has, and having that depth of expertise is helpful when encountering new programs and molecules. So brief history of the company. Been around in some capacity for about 70 years. Started as a department of Ferring and Malmo manufacturing peptides. Polypeptide Group, as it is now, was founded in 1996 with the first manufacturing site opened in Torrance, which is where I work and where we do our small scale and neoantigen manufacturing. Throughout the years, we acquired several additional sites, one in France, San Diego, which also does smaller scale manufacturing, a site in Malmo, opened one in India, Lonzo's peptide business from Belgium, and then most recently in 2021, we IPO'd on the Swiss stock exchange. So that puts us at about 1,200 employees worldwide as of June 2022. Brief breakdown of sort of who's working here. Pretty even spread of people who joined quite recently and people who have been here for 10 plus years. So our development group has a decent number of highly educated individuals, and that sort of extends into our production line. A lot of what we do is custom manufacturing, especially on the peptide side. So we want to make sure that the staff we've got running those operations is able to handle problems quickly and efficiently. So a lot of our production staff do have academic degrees. Breakdown of where we're located, we do have 6 manufacturing sites across 3 continents, 2 in California, 3 in Europe, and 1 in India. There's our executive team in Switzerland because that is where the company IPO'd. So a history of NAPP specifically. We were first approached to do some of this neoantigen or personalized manufacturing in 2015. At the time, the board didn't like the idea. So as much as we wanted to start on this work, we weren't able to. Luckily, the market moved a little bit, and some of more programs were out there, and we sort of had the opportunity to say, okay, maybe there's more than one client out there for us, which is important as a CMO. And so 2018, we were able to capture one of these pilot programs. And initially, this was being performed in a development suite. The instrument pictured here is not something we ever actually used, but it sort of captures the how well organized our equipment was to start off with. A lot of what we were doing resulted in fairly long turnaround times and not particularly high throughput, just because we hadn't designed the space for exactly what we needed it for yet. We were using the space we had, and often you'd get about halfway through a project, find out that there was a small error with your equipment because you were still getting used to it, have to go back and repeat maybe 10 peptides, and that process really adds a lot to the turnaround time. Around the same time, we expanded our production facilities. I think we had 2 sites in 2018, 2 buildings dedicated to manufacturing. That has since jumped to 4, and with that space increase, we've been able to capture a room specifically for small-scale peptide manufacturing. And so this is what that suite looks like now. Much better organized equipment, a lot of more space, cleaner environment for actual manufacturing. So we do now have a dedicated GMP suite and personnel for small-scale peptide manufacturing, where initially it probably would have taken us about 6 weeks to manufacture 1 order of peptides at this scale. We can now do 3+ patients a month. And depending on the scale, it could probably be significantly faster than that. I've got a few milligrams of 20 peptides in 2 weeks. If you're just looking at the manufacture portion with our QC, that can potentially be finished in as little as a week. And since the development days, we've fully engaged our quality system, so we're generating a GMP product. And then I mentioned the smaller scale, so 20 peptides in a few weeks. We've also done programs in this space more geared toward generating peptide libraries with something like slightly larger quantities of about 200 peptides. That obviously takes quite a bit longer than 2 weeks, but if you need a library of peptides, this kind of space can still generate that material. In the case of this autosynthesizer, the way we've sort of set up our manufacturing is we can do maybe 12 to 16 per shot. So you set up the instrument, hit play, it'll do it 16, which is about the right amount to set it up before you leave, have it finished for you when you come in the morning, swap everything out, have it run over like throughout the day, and then repeat that until you've generated your peptides. We are a little limited on scale for this instrument, but the advantage of having it be fully automated means that disadvantage is worth it. It's often better to run 2 at a smaller scale than try and scale it up, at least with this instrument, because it lets you just fully automate the process. I mentioned that it's GMP manufacture. For us, that is limited to drug substance. We cannot do the fill finish portion of the process, but we've managed to get a pretty efficient run through of just the peptide manufacturing itself. So some challenges that are unique to the personalized approach. First off, it's a constantly changing product, which we'll get into a little bit. Turnaround times are critical, and they need to be very fast. The longer you wait, the worse your patient outlook could become. Maybe the targets you're looking for change by the time you get it to the patient, so being quick is important. Some approaches to control strategies need to be a little bit different, given that you're not making the same thing over and over. And then when you go to increase manufacturing capacity, you can't just make it bigger, because you don't want more of what you're making. You want a wider breadth of what you're making. So constantly changing product. Every time you go through it, it's going to be a novel molecule. Just on the flow charts on the side here, the top one ideally is a developed process for a commercial product. You're able to define what your normal operating ranges are, and as long as you're staying within those, your product is likely performing the same every time that you run it, because it's the same every time that you run it. For a peptide that is 20 amino acids long, you've got something like 100 septillion possible molecules that you could be making. And that number does not mean anything to me. It's sort of too large for me to understand. And so if you were to go in and try and anticipate every possible thing that is going to happen, you would not make it to manufacturing in a lifetime. So the number of branch decisions that could come from a novel molecule is tremendous. So instead of trying to anticipate everything, our approach, and what we think is probably a more advantageous approach, is to build flexibility into your system and try and make it robust as possible. Ideally, you're not needing to diverge too often, but because it's new material every time, that is going to be unavoidable. So take a rich-based approach to that process development, stability programs, specification. Sort of two examples of this is expect a reasonable level of attrition. If you want to make everything, it is probably possible. But we've had an order of 20 peptides, and we got maybe 16 of those within a week and a half, and then had to do another week and a half of development per peptide in order to get it through manufacturing. So if that timeline is acceptable, then that could be an approach. But often the turnaround time is important, and so just accepting that some of these peptides are not going to make it into the product, and structuring an order where that can be acceptable, having a broad enough spectrum of peptides that can work, and it won't reduce the efficacy of the therapy, is pretty important. And then when you're going into sort of your process development and stability programs and things like that, developing a worst case or a expected case and doing your studies on that is important. You don't want to go in and say, well, this is what we're hoping things will look like. You don't run into any issues during engineering runs. Then you get to your actual order and you run into a bunch of new issues that you haven't built contingencies for. So you want an adaptable platform. Ideally go in with some predictive tools and leverage analytical results whenever you can. We have a system it'll predict the elution point on our analytical gradient. It is usually pretty wrong. I think it gives 14%, it'll come out at 20. And it's not consistently wrong. So this is a portion of the prediction tool that we sort of don't use, and we just use our analytical results as a great way to inform how we should be doing those purification steps. If you see it perform on your system, you can better predict how it's actually going to perform when you need to scale that up for purification of the whole molecule. So addressing critical and rapid turnaround times, our approach to this and what we think is critical to getting this done is run parallel manufacturing whenever possible. The more steps of the process you can be doing at once, the sooner you'll be getting completed with the process. And also, in the same vein, you want to hit your bottleneck as quickly as possible. Whatever the slowest portion of your process, you get there quickly and get it running so that you can finish your process. I've got an example schedule of what that might look like from synthesis through lyophilization process. And as you can see, there's about a day lag between each process. As soon as we have any material out, it is going into the next stage, and we are pushing it along as quickly as possible. This approach also gives you the opportunity, as if you do run into some issues during purification, if your synthesis process is still running, you can go back, restart that molecule, and give it another go in purification without hugely impacting your timelines, assuming that you've sort of built that into your program. From the project management side, run everything as a sprint. Once the patient order comes in, every activity needs to be tracked extremely closely. If you have a delay starting your synthesizer by a day, well, that pushes your turnaround time out at least a day. So you need to make sure that everything is closely monitored and you're pushing it along efficiently. When it comes to quality incidents, I've worked on programs where you get a deviation, you inform the client, and they take about 5 days to deliberate, provide some feedback. You work on it for 5 days, and it ends up taking about 20+ days just to sort of resolve a fairly minor investigation or deviation. That is not an option when your whole timeline is 14 or fewer days. So making sure that there's sort of alignment on quality expectations going in, and that you really focus on product impact for those deviations is extremely important. Building in attrition can be very helpful here too, because it lets you say, okay, we don't have time to appropriately evaluate this at this time. We're going to let that be part of our attrition and move forward, and then we can evaluate it when we have more time, when the patient outlook is not on the table. And so how to effectively make this happen, communication is hugely important. Yvonne mentioned this between client and CMO, being able to quickly reach each other, come to decisions together, and move forward becomes super, super important. And I'm really glad that we have such great partners who are willing to work with us and available at the late hours that we're still working, even in different time zones, to make sure that we're able to push the process forward efficiently. And I think that partnership is critical to effectively manufacturing personalized therapy. So for control strategies, a lot of this is the same as if it was, sort of any product. You want to be monitoring your environment, evaluate sort of the risk of each step for contamination of your product, and use classified environments for the particularly sensitive steps. Make sure you're tracking your raw materials and consumables in some way. ERP system is a good method for this. Where it maybe starts to diverge is the utilization of consumables becomes much more important for a personalized program. You're not able to do a cleaning study on every single molecule and make sure that it's fully removed for reusable components. So as much as you can, using consumables lets you not have that worry that something might become cross-contaminated. Similarly, operating in closed systems, our synthesizers are fully closed. Obviously, an HPLC is a fairly effective closed system. Really reduces the risk of some powder flying through the air and getting where it shouldn't, mixing of peptides, or a much worse case, 2 patients being cross-contaminated. Making sure those are closed and then as much as possible automated is really important for making sure you have clear separation of material. On the left here, we have a sort of breakdown of our room and color coding of which activity is happening in each zone, and making sure that you've got a logical flow and things are segregated in that way is important, again, to make sure people are working with the material where they should be, and you don't have the risk of accidentally mixing things up. You want to clearly label where things should be, and then make sure you're observing those labeling requirements and staying within the zones you need to be. So increasing throughput becomes quite a bit more challenging. If you want more of a generic drug or something, you put it in a bigger reactor and you make more of it. The scale up for a personalized therapy is usually not advantageous. For most of what we're doing, like the majority of the product is going to analytical testing, not actually into the product. You don't need very much for a lot of these therapies. So scaling up doesn't help with increasing your throughput, which means the focus needs to be on streamlining the process and as much as you can automating. So minimizing operator interaction. In the case of the autosynthesizers that we're using, they're capable of running 24 peptides. And so we used to just load up 24 because it seems efficient, hit play and come back a day and a half later. But that system would be idle basically the entire evening of that, the entire second evening, which ends up being much less efficient. And thankfully, we were able to rely on some of the staff working in that area, working primarily with the autosynthesizer to say, okay, what is the most efficient way for us to be running this? And they were able to devise the new scheme of making sure that it is running as much as possible. So maximizing that equipment utilization, both through the automation and smart planning of equipment use. Continuously monitoring your process. You want to know where your bottlenecks are and continuously have a plan to address them. So again, initially that was synthesis. And in our case, it's now moved down towards purification. And we're working on means of sort of getting the operator out of that process and having it run itself a little bit more than it currently is. And then this sort of unrelated to scale up, but scale your investment with demand. You want to make sure that you're spending appropriately based on how much your expected need is. You don't want to spend millions of dollars generating this high-tech automated system if the amount of material needed is going to be something you can manage with a NAND process. And so I think my hope for where this is heading is as the science gets proven, automated processes will take over more and more, hopefully driving down cost and making this an accessible therapy for folks. Because at the current price point, insurance companies are great, but it is a little aggressive in terms of costing. So as it gets proved out, I'm optimistic that automation will meet those needs and reduce cost while making the scale manageable. So once you've sort of been limited by your ability to streamline and automate systems, that's when you might have to look at scaling out, take up more footprint and just do more of it. Yeah. So advantages of partnering with a CMO. This is the brief shill. It allows the investment in terms of manufacturing to be business driven. It takes away the need to set up your own quality system, to hire individuals with expertise on GMP and manufacturing, and then creates flexibility on sort of how much you're paying for that staffing based on the demand of the program and an expected number of orders. Setting up a manufacturing organization is not particularly cheap. And so if you've got a limited number of orders initially, it likely doesn't make sense to make that investment initially. And then it lets you sort of make continuing decisions based on performance of those clinical trials. If things are going well, great, we'll scale up with you. If things aren't going as well, well, you don't have to shut down a new business unit. We are able to find alternative work for that space and make it grow. Also, you get a effective partner leveraging, in our case, experience with NAPP production, 20 plus years of regulatory experience specific to peptides, global quality system, qualified raw materials, and established supply chains. Finding raw materials can be a lot of work, and we've already done that portion for you. So go with the CMO. Big thank you to the PPL team working on neoantigens. Without this team, I would not be here today, and we would not be able to make this material. So huge thank you to the Polypeptide both quality and manufacturing team. now have time for questions.
Okay, thank you. Thank you, Cary. So we'll take any questions again, just like before. If you have any questions, you can type them in the Q&A section, or you can raise your hand and we can bring you onto the stage to ask the question. So any questions from the audience? So Cary, in the meantime, while we wait for questions, similar to the question I asked Yvonne earlier, going through this process, what would you say are some of the key learnings that you learned as you established the process and worked with different clients and developed the process, if you will?
I hope I'm not just stealing Yvonne's answer, but effectively collaborating with your partners is extremely critical. I think both from the CMO side and going both ways, being receptive to feedback and taking that into consideration and adjusting your approach as barriers come up is super important. You don't want to go in with a strategy and say, I'm convinced this is going to work, which I've been guilty of. But you want to make sure that you're flexible with how you're approaching problems.
Okay. We do have a question from an M. Neely. It says, is there interest in or a need for a facile and flexible formulation technology that would expedite the movement of personalized peptide products from synthesis to patient or caregiver?
I don't think I really have an answer for that as the peptide CMO. We have worked with, or at least recommended, fill finish sites for our products. So there is a need to perform the sterilization and potentially formulation of this material. But in terms of from the polypeptide perspective, I don't really have a response on that one.
Okay. Walid, I see that you have a question. You said a startup company, Boston, in collaboration with MIT scientists, developed a peptide program. Are you referring to my type, Walid? Yes, so that is my type. And I think they presented at the Boulder Symposium last year. But I think the scales at which they operate are quite small relative to what is typically requested, and also the GMP compliance, which was a question that was asked from Yvonne's session, is also important, which doesn't get addressed at this point, at least.
Michelle, I can't ask a question in the chat. Do you mind if I ask a question?
Yes, please.
Okay. Cary, so from the CMO perspective, what do you think is the biggest challenge in individualized peptide manufacturing?
I think two components. The scheduling can be challenging. I know our partners do generally a pretty fantastic job of making sure that we have visibility on expected orders, and then I also understand that is because it's a clinical enrollment that it relies on that changes a lot. But sort of working with a regular commercial product, you set your schedule at the beginning of the year, and hopefully you stick to it. With a personalized program, you make it when it's needed. And so organizing a schedule around that can be difficult, and I sort of mentioned it already, but the collaboration there is super important to making sure you have an effective system. I know I said there's two parts to it, but I don't remember what my second part was, so that's going to be my whole answer.
Thanks.
there any other questions? So if there are no more questions, I think we can call this a day for the for this seminar. But Lauren, I believe you want to talk a little bit more about the next session, next seminar?
Yeah. Brian, do you want to? I think Brian's going to come on and wait for him to get started. Going to give a little plug for the next virtual seminar.
Sorry, I just had to restart, so if I missed something, yeah. So in May we'll be doing our next webinar. The focus will be on peptide immunogenicity. So we hopefully have a speaker from one of the labs at the FDA who will be talking about innate immune response, which is one of the things that the FDA is now asking people to look at, and how that could impact immunogenicity and safety of the peptide. And then I'll be talking about in silico and in vitro methods for evaluating direct peptide immunogenicity and T-cell response, which is another part of the application for both generic and novel drugs, and importance of certain features to look out for when you're doing those evaluations.
And it'll probably be early May, right?
Mm-hmm. Within the first 2 weeks.
So you can watch for some advertisements for that. I'm glad everyone joined us today. Thank you, Cary and Yvonne, for your great talks. Becky, did you have any closing words or?
Yeah, just thanks to our two speakers. It was a really great topic, and thanks for everyone who joined us. Look for our live meeting in September too. That's coming up too. So look at the agenda and hopefully join us in NAPP. Thanks, everyone.
Thank you.
All right, thanks.
Thanks.